Cell Biology Dungeons and Dragons. Cell biology was a pretty basic course that many of our school friends had to get through. Those friends included a group of people we played Dungeons and Dragons with. An eclectic collection of acquaintances, schoolmates, neighbors and workmates often inhabited our basement for a 3-day D&D session. Pizza and chicken boxes had to be bulldozed out the double doors along with a mountain of sleeping bags, blankets, and pillows. It was an ill wind that blew through our neighborhood on those mornings. Several stuffed animals still remain in our lost and found. It really wasn't that difficult to find these people at Oxford or in Edinburgh in general. If you walk around on a sunny day and observe the people under trees reading books or hiding behind poles acting like they're trying to look indiscreet, then you have the foundation of your group. Our group included a girl by the name of Shelley. That wasn't her real name, but the name of her current persona, which was to live in Shelley's world. She dressed in a Victorian modern kind of way that was unique to her. Shelley was the perfect dungeon master because with her knowledge of the game as well as her knowledge of cell biology only she could create this session. In cell biology we studied the structure, function and behavior of cells including both prokaryotic and eukaryotic cells. Prokaryotic cells do not have a cell nucleus or other membrane-bound organelle. They are the smallest form of life and include bacteria and archaea which both reproduce through binary fission. Eukaryotic cells can either be unicellular or multicellular and include animals, plants, fungi, and protozoa. The test we all had to pass was based on the components of the eukaryotic cell. Shelley devised a D&D session to help us remember those components. Normally people who didn't have to pass the test could care less about this information. In D&D, if you get to play a character, you're part of it. Everyone got to play at least one character from the cast of the nucleus, the nucleolus, the endoplasmic reticulum aka Miss Mitochondria, the Golgi apparatus, the lysosome, the cytoskeleton, the cell membrane, centrioles. The nucleus was played by Simone Westerlake, a student in our year but not in many of our classes. The only other connection with her was when she dated Norbert about a year ago. The nucleus was the lead role and stores the genome and genetic information for the cell, containing all the DNA organized in the form of chromosomes. Then the journey begins. Its armor is a nuclear envelope that has nuclear pores to allow the transport of proteins inside and outside of the nucleus. This is key to passing changes. The replication of DNA as well as the transcription of DNA to RNA must happen here, and this is the first hurdle in the dungeon quest. Afterwards, the RNA is modified and transported out to the cytosol to be translated to protein. The nucleolus was also played by Simone Westerlake by holding out her arms. The nucleolus is inside the nucleus and is dense and spherical in shape. It deals with ribosomal RNA, rRNA, synthesis required for ribosomal assembly. The endoplasmic reticulum was played by Wild Bill Kronolak. Wild Bill earned his name many times over, mostly through his bar exploits, but he was always a great guy to run into at any bar. He could bring most nerds out of their shells and get them in more trouble than they've ever been in. This character suited Wild Bill's personally to a T. Commonly known as this structure is a network of membranes found throughout the cell and connected to the nucleus. A real hanger-on type that likes to deal in information. The membranes are slightly different from cell to cell and a cell's function determines the size and structure of the ER. A very adaptable guy, perhaps even malleable. ER synthesizes, stores, and secretes proteins, then deals them off to the Golgi apparatus. Several missions and major quests had to be executed to build a network with the Golgi. Tricky buggers, the Golgi. Miss Mitochondria was played by Nadine Patterson, the current saddle partner of Wild Bill, but Nadine was also in our course and that's how Wild Bill met her. Miss Mitochondria produces energy in the form of ATP within the cell. Her homeland hosts the Krebs cycle or TCA cycle were dedicated to the production of NADH and FADH. Those products are used within the electron transport chain, ETC, a non-player character, and oxidative phosphorylation for the final production of ATP. The Golgi apparatus was played brilliantly by Errol. The Golgi functions to process, package, and secrete the proteins to their destination. Protection is necessary. The proteins contain a signal sequence which allows the Golgi apparatus wise guys to recognize and direct it to the right place. Errol played this role brilliantly in an Al Capone personae. I played the lysosome less brilliantly, but hopefully a competent performance. The lysosome functions to degrade material brought in from the outside of the cell or old organelles. This contains many acid hydrolases, proteases, nucleases, and lipases, which break down the various molecules. As they attacked me in various quests, I was able to fight them off with acids and other vile liquids and gases. My specialty was autophagy, which is the process of degradation that occurs when a vesicle buds off from the ER and engulfs the material. Then it attaches and fuses with the lysosome to allow the material to be degraded. It's a head-to-head match against Wild Bill, and he called me horribly insulting names. The ribosomes were played by Raj Kanwar, a friend of Wild Bill's. The ribosomes translate RNA to protein. It serves as a site of protein synthesis. This is a quest that must be made when Raj shows up over the timeline of the D&D session. Let's just say Raj moved on his own timeline. The cytoskeleton was performed adequately by our neighbor Sonia Swan. Sonia was an accountant at a local company and had recently purchased the house next door. Errol was helping her get settled in and invited her to join us. She had never participated in anything like this before and was skeptical, but we had a role that needed filling. I'm sure she had it in the back of her mind that we were some kind of cult. Sonia was cytoskeleton, a structure that maintains the shape and general organization of cytoplasm. She must also anchor organelles within the cells, creating structure and stability. Cytoskeleton is composed of three principal types of protein filaments or henchmen known as actin filaments, intermediate filaments, and microtubules. They're held together and linked to subcellular organelles and the plasma membrane by a variety of accessory proteins, which is one of the most difficult quests on Shelley's journey. In retrospect, a more familiar player would have been better in this spot, but in reality, Errol also played this part through Sonia. Cell membrane was played by Sonia's boarder Dominic Balagumba, a medical student. The cell membrane can be described as a phospholipid bilayer which consists of lipids and proteins. Because the inside of the bilayer is hydrophobic, and in order for molecules to participate in reactions within the cell, they need to be able to cross this membrane layer. Spells for this character include osmotic pressure, diffusion, concentration gradients, and membrane channels. Awesome. Dominic played this part well, and as a medical student knew his material well. Centrioles was played by Dominic's friend Igor Ruzmoinets. Igor worked in a tattoo and piercing shop near the school that was quite popular with the first-year students. Cheesy would have called Igor a nip and scrote guy. The centriole's function was to produce spindle fibers which are required to separate chromosomes during cell division. Eukaryotic cells may also be composed of chromatin to make up chromosomes and a mixture of DNA with various proteins as well as cilia to propel substances or for sensory purposes. I don't think Igor was a student, but I didn't ask either. He did say to Dominic at one point, "I thought you were talking about cells with bars on them or something." That was the most memorable D&D session I was ever a part of. Great job by Shelley.